The myogenic mechanism is going to involve smooth muscle, because myo means muscle. The myogenic mechanism is something that occurs in other places in the body too. And basically, this is just referring to the tendency of vascular smooth muscle to push back when it's being pushed. Let's take a look at how this applies to the kidney. If the afferent arteriole is under high pressure, if it's being stretched, then the smooth muscle in the wall of the afferent arteriole will push back, meaning we're going to get constriction of the afferent arteriole. This will prevent really high blood pressure from entering the glomerulus and possibly damaging it. And to be honest, with every beat of the heart, we experience a high pressure followed by a low pressure. And if that pressure is too high, it could damage the glomerulus. So this myogenic mechanism is really important in regulating what's happening at that nephron, at the nephron level. The decrease in the afferent arterial diameter is going to cause a local decrease in glomerular filtration rate for that particular nephron. The opposite is also true, meaning the afferent arteriole is going to dilate when it's under low pressure. So again, with every beat of the heart, we know that during ventricular systole, the pressure coming into arteries is high. But when the heart is undergoing diastole, the pressure in those arteries will be low. So the afferent arteriole is going to dilate when it's under low pressure. How do you think dilation of the afferent arteriole is going to affect that local glomerular filtration rate? Is it going to increase it or decrease it? If you said increase, that's correct. Because if the pressure is really low coming into the glomerulus, it might be too low to perform the process of filtration. So by dilating that afferent arteriole, we can increase the pressure in that glomerulus just a little bit, ensuring the process of filtration continues despite that low blood pressure. We can also use the juxtaglomerular apparatus in the process of autoregulation. We've already looked at how we can use the juxtaglomerular apparatus to cause a release of renin and activate that renin-angiotensin-aldosterone system. This is kind of related. So we're still going to use our macula densa cells. Don't forget, those macula densa cells can detect the flow rate in the distal convoluted tubule. But in addition to detecting flow rate, they can also detect the fluid composition. They're especially sensitive to sodium, but they are sensitive to overall osmolarity. If the sodium concentration or overall osmolarity in the renal tubule is too low, and typically that happens when the blood sodium and osmolarity is too low, then the macula densa cells release chemical signals that will cause the JG cells to contract. The JG cells are a type of smooth muscle cell that wraps around the afferent arteriole. So when the JG cells contract, the diameter of the afferent arteriole decreases, which will lower the pressure in the glomerulus, decreasing glomerular filtration rate, and decreasing that flow rate too. This is going to lower the GFR, and by lowering the GFR, we're going to give the nephron more time to reabsorb sodium. And by reabsorbing more sodium, we can increase our blood sodium levels, which will then increase our tubular fluid sodium levels, and then we will deactivate this system. Nervous system regulation of urinary function is way less complicated than using the endocrine system, so you can breathe a sigh of relief. We're going to use the sympathetic nervous system to regulate urinary function. There is no parasympathetic signaling to the kidneys, just sympathetic only. We know our sympathetic nervous system is our fight or flight, our emergency nervous system. And when the sympathetic nervous system kicks in, one of the things it's going to do is to send signals to the kidneys, increasing constriction of the afferent arteriole. We're mostly doing this not for the sake of the kidneys, but for the sake of the rest of the body. By constricting the afferent arteriole, we can redirect that blood to organs that really need it right now. Who cares what's going on in your kidneys when there's an emergency going on? Your brain needs that blood. Your heart needs that blood. Your skeletal muscles need that blood. So we're redirecting the blood elsewhere. Consequently, this will cause a decrease in glomerular filtration rate because we have less pressure coming into the glomerulus. Now there is a little selfish thing happening here for the kidneys though. By constricting the afferent arteriole, we are protecting the glomerulus from high blood pressure. When that sympathetic nervous system kicks in, we're going to increase respiratory rate, increase heart rate, and increase blood pressure. And that high blood pressure could potentially damage the glomeruli. So by constricting the afferent arteriole, we're going to decrease that pressure coming into the glomerulus, protecting it. We will also activate the sympathetic nervous system to the kidney if blood pressure becomes too low. If blood pressure is too low, the nervous system can actually stimulate the JG cells to release renin. Now, we know that renin ultimately leads to the activation of angiotensin II. And angiotensin II, what it did was it caused both the afferent and the efferent arterioles to constrict. It caused the afferent arteriole to constrict a little, but it caused the efferent arteriole to constrict a lot. And what that meant was we had more blood coming into the glomerulus and less blood coming out. So we could build up a bit of pressure in this glomerulus despite the fact that we had low blood pressure. When that blood pressure is too low, we can't filter blood. So we can use renin and hence angiotensin to maintain the glomerular filtration rate despite the fact that we might have low blood pressure. Of course, the angiotensin II is also going to correct the whole situation by causing an eventual increase in blood pressure back to normal.